This podcast is going to walk you through cell reproduction. We're going to look at prokaryotes, which include things like bacteria, and then we're going to focus on eukaryotes, which are plants and animals. So quickly looking at prokaryotes, just a quick definition. Prokaryotes are organisms that lack a nucleus. They have single chromosomes, and they will reproduce primarily by binary fission. They include things like bacteria. And some steps in binary fission, this is used by bacteria. The cells will increase their cell mass slightly. The DNA and cell components are replicated, and each cell divides into two daughter cells. So this is a bacteria here undergoing binary fission. They do make an identical copy of themselves. These are E. coli, and again, they're dividing by binary fission. You can see this one here. There's some more here dividing, making identical copies. Now eukaryotes, on the other hand, they contain a nucleus. They also contain membrane-bound organelles. Eukaryotes do have asexual reproduction in cells, and that's done through a process called mitosis. And the rest of this podcast is going to focus on mitosis and the events of mitosis. Before we get into mitosis, though, we should talk a little bit about the cell cycle. The cell cycle is divided into stages. Now, traditionally, we've always understood that we have the M, or the mitosis stage of the cell cycle, and the rest we call interphase. And in fact, interphase is divided into three different stages. It's divided into the G1, the S phase, which you'll remember from the previous unit we did on DNA replication, and the G2 phase. Now, cytokinesis is in there, and cytokinesis is just a word for cell division. So looking at some of these stages, during the G1 phase, this is where the cell will start to grow. It increases in size, and it prepares to copy its DNA at this point. During the synthesis phase, we've already talked about DNA replication and how DNA replication occurs. Well, that's where this will happen in the synthesis phase, and the chromosomes are essentially duplicated. The G2 phase is between the synthesis and mitosis. The cell continues to grow, and it makes proteins needed for cell division. Now, during the M phase, obviously the mitosis phase, this is where the cell's energy is used to divide. It divides into two daughter cells. This is also called karyokinesis, nuclear division. So the life cycle of a cell, I wanted to include this just to remind you that mitosis is a cycle. And so we'll be looking at stages of mitosis, almost like they're pictures, but I want you to remember that this is a continuous cycle that occurs. It has no beginning, it has no end. And we use the stages mainly as an easy way to recognize key points in that cycle. So starting with interphase, the resting stage, this is where the cell will carry on normal activities. During this stage, the chromosomes aren't visible, cell metabolism's occurring, and this is before mitosis. Now also remember, DNA replication occurs, cell growth occurs, protein production occurs, and this is what we term interphase. So here we see a diagram of cells in interphase, and you can see the nucleus of each of these cells, and there's no real chromosomes visible. You can see the nucleoli of some of them are quite visible, and that's what interphase would look like. Mitosis itself has four main stages, and again, they're only pictures of a continuous cycle, and those are prophase, metaphase, anaphase, and telophase, and we'll go through each of those really quickly. Before we do that, we should look at some chromosome stages and chromosome numbers. So if this was a sample cell, our mother cell maybe would contain 10 chromosomes. Remember, prior to this, during DNA replication, the DNA material is doubled to, this was 10 chromosomes, we would now have 20 chromosomes worth of DNA material. And finally, after mitosis takes place, we would end up with daughter cells that have 10 that are identical to the mother cell. What would this look like in humans? Well, in a regular human cell, we would have a diploid 2N cell with 46 chromosomes. DNA replication would double that to 92. or a tetraploid cell, a 4N cell. And then after mitosis, we would have two daughter cells, both with diploid numbers again. To go over briefly the stages, and then we'll look a little closer at them. Prophase is where chromosomes become visible. Metaphase, they essentially line up, and in anaphase, they separate the chromatids, and in telophase, we end up with cytokinesis occurring, the cytoplasm divides, and the nuclear reform. So that's sort of the overview. Again, those are the stages. This is an image of an onion root, and you can see that there are several cells where mitosis is taking place. There's a nice metaphase one there and there. You can see some prophases occurring. Now, steps in prophase. First of all, the DNA will become coiled very tightly. Remember, the DNA is very loose and it's invisible in interphase. It becomes very tightly. The nuclear membrane disappears, nucleoli disappear. The centrioles will begin to migrate to the poles. That's only in animal cells. Plant cells don't have centrioles. And the spindle will begin to form. So this is a cell in prophase, and you can see the DNA material is now packed into what look like chromosomes, kind of look like spaghetti. A couple chromosome terminologies to sort of keep in mind. If we look at a chromosome here, the centromere is what would hold the chromosomes together. This, by the way, is a chromosome. This is a chromatid, and we have two of them. And remember, we get those because of DNA replication. DNA replication makes an identical copy of that chromosome. And then located on those chromosomes, we'd find genes that would code for traits. So two identical chromosomes are called homologous pairs, and identical chromatids are called sister chromatids. So if this was on a human chromosome, this could be chromosome 1, which would have a locus for a particular gene, and they may be different in terms of their forms of that gene. But that's sort of some of the terminology, and we'll be discussing this a little bit more in genetics. So these are human chromosomes, and this is obviously a karyotype, and a karyotype looks at chromosomes in humans. Chromosomes are typically labeled 1 to 22, and then there are the sex chromosomes, X and Y. So this, because there's an X and a Y, this would be a male. Chromosomes are numbered based on their size. So they go from the longest chromosomes all the way down to the shortest, and then the sex chromosomes are afterwards. Going to metaphase now. In metaphase, the cell prepares to separate the chromosomes. So the cell will actually align the chromosomes in the middle of a cell. And so this is a cell in metaphase right here. And you can see the chromosomes are aligned in the middle. In anaphase, the chromosomes separate. And they do this by the spindle fibers shortening. So the chromosomes are pulled to the ends of the cell. And this is a diagram just showing us basically that we are pulling apart, or the mitotic spindle is attached to those chromosomes by their centromeres and will pull apart those sister chromatids. So here's a nice cell here in anaphase. You can see the chromatids are being pulled apart. Telophase is the last cell. It's the separation of the chromosomes. The cell plate, if we're a plant, will begin to form. If we're an animal, we don't have a cell wall, so a cleavage furrow forms. The nucleus and the nucleolus will reform, and chromosomes will begin to uncoil and become invisible again. So here's telophase in a plant cell. You can see this right here. That's the cell plate. And in an animal cell, obviously, you can see the cleavage furrows, and the cell will begin to pinch off. Cytokinesis is sometimes occurring alongside a telophase, and oftentimes people put them together as one event. It's basically the separation of the chromosomes afterwards, and this forms the two identical daughter cells. So here's a plant cell undergoing cytokinesis, and again, it's very hard to tell this from telophase, and a lot of times it's identical or it's exactly the same. So that was the note set on mitosis.